Hey everyone, today's webcast is going to be about nervous tissue, so we'll get right to it. Two main cell types of nervous tissue are going to be the neuron, which is the excitable nerve cell, which people classically associate with the nervous system. And there's going to be a second main type called neuroglia, or sometimes just glial cells. You'll hear it referenced like that. And these cells surround or wrap around neurons, and they act as support cells. If you identify transmitting a signal as the main job of the nervous tissue. So we'll go over some parts of the neuron here. First, there's going to be two parts that are similar to other cell types. It's going to be the cell body. Sometimes is referred to as the soma. And then the nucleus. You're familiar with those two portions from other cells, and there's going to be some cytosol within there. Now here we're going to have a unique structure to neurons, and this is called a dendrite. This is what's going to receive signals from other neurons. They're going to create synapses and send it to the cell body. Then we're going to have the axon hillock. I'm not going to get into too specific details about what everything does, but this is where an action potential is going to start from. This is where it's going to generate, and if there's enough excitatory input on this nerve cell that reaches the axon hillock, you're going to get an action potential, and this nerve is going to fire, and you're going to get a signal to go down the next part of this, which is the axon. We have the axon here. And then finally at the end, we're going to have the axon terminal. And this is going to synapse onto dendrites of the next cell to create that communication and release neurotransmitters. Axon terminal. So there's some other parts here that aren't always present. So we're going to have, I'll draw in myelin, and this is an insulation around the axon. What that's going to allow is faster signal transduction. Myelin, hope I spelled that right. And I'll draw a second one here. Myelin. And what that's going to create is this node of Ranvier. And that's going to be the gaps between these individual myelin units. And as a quick note, when there is myelin, and what makes it a faster transduction of signal is something called saltatory conduction. So myelin allows for saltatory conduction. Abbreviate that. And the saltatory comes from the Latin root saltus, which means jump. So saltus, that's where the saltatory conduction comes from. And so what happens is when you get a normal action potential, it's going to travel, it's going to depolarize along the cell, all down every last square area or unit area of the axon. When you have myelin, it's only going to depolarize here, and it's going to depolarize here, and it's going to depolarize here, and depolarize here. So you only have these 4 locations in this situation instead of having the entire length of the axon. And that is going to jump from node to node, and that's what that saltatory comes from. It's probably too much detail for this webcast, but that's just to give you some background. So that's going to be the general anatomy of a neuron. The next thing we're going to talk about here is, I'm going to leave this up here for the most part. We're going to go over some nomenclature, or how things are named, or what I'm referring to when I'm saying a certain word. So nomenclature, just so there's no No confusion. First we're going to have a neuron, which I defined above. We have a neuron. It's going to be base excitable nervous tissue. Base excitable nerve cell. The next thing is a nerve fiber. A nerve fiber. What this is going to be is a bundle of neurons, and they can be going both ways. Bundle of neurons in the peripheral nervous system, specifically. In the peripheral nervous system. And that's going to differ from a nerve tract. A nerve tract, which is going to be the same functionally, but found in the CNS. The central nervous system. So it's going to be the spinal cord or the brain. It's going to be everywhere else. So nerve fiber, bundle of neurons in the PNS. Nerve tract, bundle of neurons in the CNS. The next thing is going to be a ganglion. Ganglion, that's an A. This is going to be a grouping of cell bodies in the PNS. Grouping of cell bodies in PNS. And then we're going to have an analogous term called a, let's see, for this we're calling it a brain nucleus, and sometimes it's just referred to as a nucleus, which is incredibly confusing, but we'll call it a brain nucleus for this situation. Nucleus. And this is going to be a grouping of cell bodies in the CNS. These are very, very important to understand because they could completely change the answer to a question. And if you're confused with any of these, especially this brain nucleus, it's going to make the rest of the material pretty confusing. Alright, now we're going to discuss how a nerve is an organ. So we've talked before how an organ has to have four main types of tissue. How much space do I have here? So we're going to talk about how the nerve itself is actually going to become an organ. The nerve as an organ. So if you think back to when we talked about muscles, we talked about how they're organized within each other and wrapped around with connective tissue. A nerve is going to have that same organization. That's going to be, let's see here. I'm not going to draw the whole thing like I did out with the muscles. I'm just going to draw parts of it here. Okay. So imagine that we're looking down, looking longitudinally along a nerve fiber. Longitudinally along a nerve fiber. Here we're going to have our epineurium. Epi, we have that epi prefix again, just like the epimysium in muscle cells, in muscle tissue. Epineurium. This green, then, if any of you can remember, you could guess what this is going to be called. Perineurium. And then this purple, endoneurium. Endoneurium. So it's organized exactly analogous to how the muscle was, where we had the epimysium, perimysium, and endomysium. So that should be easier for you to remember. You have epi, peri, and endo again, and so neurium is going to be the second part of that word. And then within that endoneurium, this is the neuron itself. And that could be running in either direction. So these three, so the nerve is an organ, we described its gross anatomy there and how it's organized. What we have here then is our connective tissue. Connective tissue. We have our nervous tissue, obviously, in our neuron. Ooh. Nervous tissue. Let's see what else we need here. Originally, we talked about always having muscle and epithelium. Connective tissue, nervous tissue, muscle tissue, and epithelium. I'm only going to describe epithelium because that's the only thing your book describes, and I don't want to get into too much detail if your book's not going to clarify what I say. So the last part is our epithelium, which we already talked about. I haven't talked about yet. The supporting cells. So connective tissue, nervous tissue, epithelium. Is going to be found in the neuroglia, called an ependymal cell. Ependymal cells. And this is what's going to filter blood to create cerebral spinal fluid. Cerebral spinal fluid. So we got 3 of those 4 main types of tissue. Like I said, I couldn't find it in your book where it goes over where muscle is, so I'm not going to talk about it. But this is how the nerve is going to be defined as an organ. Finally, so I can end this and get off here, we're going to go over the different types of glial cells or neuroglia and where they're found. Go back to green here. Neuroglia. So first we've got CNS. We're going to find there are astrocytes. Astrocytes, which the function isn't really known. But we're going to call them star-shaped. So we'll go over the shape. Star-shaped. They wrap around the dendrites. Star-shaped. And they help with neurotransmitter clearance. We'll leave that as its main function here. Then we've got microglia. And these are going to be most abundant, the astrocytes. Microglia are going to be least abundant. And they're going to be phagocytes. So they're going to engulf things, and it's going to show immune function. Immune cell for the CNS. Then we have ependymal cells, which we just went over up here. Ependymal cells. So I won't talk about that again. That's going to be epithelial. And then oligodendrocytes. Oligodendrocytes, which create myelin sheaths for the CNS. So there's 2 types of neuroglia found in the PNS, and I'm just going to draw them up here and then draw arrows to because they're going to be similar to ones found in the CNS. And I've ran out of space here. So neuroglia in the PNS. Ooh. We're going to have satellite cells. Satellite cells, which are analogous to astrocytes, but found in the PNS. And then we're going to have Schwann cells. Schwann cells, which are analogous to oligodendrocytes. Oligodendrocytes, and create myelin in the PNS. Alright, thanks for watching, guys, and talk to you in a little bit.